Hi all, I am Siddharth Subramani from Spire Lab at the Department of Electrical Engineering at the Indian Institute of Science in India. Today, I am excited to share our work titled Automatic Classification of Volumes of Water Using Swallow Sounds from Cervical Auscultation. This work has been accepted at ICASSP 2020 for poster presentation under the topic of Biomedical Signal Processing. In this work, we aim to perform volume classification through an extensive set of experiments by finding out joint features that maximize the classification accuracy. This will be the flow of this presentation. In this presentation, we will start with some insights about the physiology of the process of swallow and also look about clinical assessments for swallowing disorders. We will then move on to see about the characterization of swallow sounds and then delve into our methodology of swallow volume classification and then look at the results section. In the mouth, the voluntarily chewed food blends with the saliva to form the food bolus. Swallowing is the process of movement of this food bolus from the mouth to the esophagus through the oropharynx and the hypopharynx. This process is in sync with respiration since the pharynx is the common opening to both food and oxygen. This complex process is controlled by around 30 muscles coordinated by the cortical areas of the human brain. When the food travels through the oropharynx, the airway is protected by the muscles of the respiratory tract, which otherwise can lead to pulmonary aspiration. Swallowing disorders, generally termed as dysphagia, have been found to occur in patients who have ailments like Parkinson's disease, muscular irregularities in the esophageal and pharyngeal regions, amyotrophic lateral sclerosis, head and neck cancer, and etc. Screening tests by speech pathologists serve as a common ground for further evaluations on dysphagia. Common clinical swallow assessments include methods like video fluoroscopy, fiber optic endoscopy, surface EMG, cervical auscultation, and sometimes ultrasonography. Among these methods, cervical auscultation is non-invasive and poses no health hazards like exposure to harmful radiation and is also less expensive. Such analysis of swallow signals can find their significance in various applications like in wearable monitoring of ingestive behavior devices that detect and characterize the intake of food. In cases of acute stroke, identification of dysphagia through spontaneous swallowing frequency analysis has shown potential as a screening protocol. Apart from these, the volume-specific models can help study the level of severity of dysphagia. Now, cervical auscultation represents the action of swallow as signals. This makes it necessary to understand the signal and its different components. The previous works characterize swallow signals broadly in two specific ways. One, using swallow components, and two, using swallow sound waves. Honda et al., using a synchronized experiment of acoustic and video fluoroscopic study, concluded that one swallow wave corresponding to each swallow phase, that is, oral, pharyngeal, and repositioning phases, were found. Morinari et al. Characterized swallow signals using three swallow components and the intervals between each swallow component. But so far, no analysis has been put forth where swallow signals have been characterized using acoustic features that are volume independent. This is evident from this graph where it can be seen that the swallow components vary with bolus volume. This suggests that it could be difficult to work with swallow signals in a volume independent manner. Hence, the objective of our work is to learn features for swallow volume characterization at different volumes through a dry swallow versus non-dry swallow classification for different volumes of water. For the collection of swallow sounds, data was recorded from a total of 56 subjects. Each subject was made to swallow water of volumes 2 ml, 5 ml, and 10 ml. Apart from these, the subjects were also made to perform dry swallow. During dry swallow, saliva might have been swallowed. The cervical auscultation device used to record the swallow sounds can be seen in this figure here. This device consists of three components, namely a lifeline pediatric AL stethoscope fixed over the cricoid cartilage encircling the trachea. The second component is an acoustic tube. The third component is a Sorellas portable microphone. This setup picks up sounds from the person and PRAD software was used to record these sounds. Moving on to our proposed study, in the literature, we find that swallow signals have been described and characterized using simple temporal and spectral features. Combining all such features, we get 12 features. This set is referred here to as the baseline feature set. It consists of duration features of each swallowing sound wave, swallow components, intervals between each swallow component, duration to peak intensity, and total duration of the whole signal, and spectral features of peak intensity in each swallowing sound wave and peak intensity of the whole signal. Since these features are specific to different components in the signal, this requires manual annotation of the swallow signals by a clinical expert. We also consider another rich feature set of low-level descriptors and various functionals to investigate whether several temporal, spectral, vocal, and source features and their functionals provide cues for volume classification. This is the Compare 2016 feature set. This consists of 26 feature groups, adding up to a total of 6,373 features. These features do not require any manual annotation and are extracted from the whole of the signal. This feature set is termed here as the OS feature set. The OS feature set can, however, be broadly classified into two groups A and B, voice source related and glottal excitation related groups, respectively. The table here gives a summary of the OS feature set. Even though this feature set is more specifically designed for speech analysis purposes, this set has shown success in non-speech applications since it captures even wider spectrotemporal features from the signals. For volume classification, three classification tasks were performed, namely dry versus 2 ml, dry versus 5 ml, and dry versus 10 ml. The classifier used for this purpose was a linear kernel SVM. The optimum C parameter was selected using a grid search method based on the model performance on the validation data. A 10-fold cross-validation setup was used. The whole dataset was split into train, validation, and test set in a 70-20-10 fashion. Due to unequal number of swallow recordings in each volume category, F-score was chosen as the evaluation metric. Statistical tests were performed using the Wilcoxon signed rank test or zero median to compare the F-scores between the feature groups. We carried out 3 different studies to find out the best performing features for volume classification. In the first study, we compared the overall performance of baseline features referred to as BLF against that of the OS features referred to as OSF. In the second study, the performance of the two subgroups of OSF, the vocal tract based and the glottal excitation based features were assessed. Following this, the performance of each low-level feature category with all its functionals in OSF was assessed. Even though each category of features were assessed in study design 2, it is important to understand the minimum set of joint features that can help in volume classification. Since it is computationally very expensive to use forward feature selection algorithm over a set of 6,373 features, the lasso feature selection algorithm was used for finding the best set of features that maximize the classification performance. Since the feature rank for each fold and each task can be different, the features were selected in 2 ways. 1. Features common to all folds but separately for each classification task, and 2. Features common across folds and across classification tasks. Finally, the top 12 features equal to the number of baseline features were selected from each set and are referred to as OS ranked and OS common feature sets respectively. Moving on to the results section, results of study 1. The numbers in this table show that OSF perform much better than BLF. The p-values indicate that the difference is statistically significant from BLF. It can also be observed that when the volume changed from 10 ml to 2 ml, the model exhibited a relative drop of 46% in the F-score when using BLF, but the drop was only 9% when using OSF. This is an indication that OSF is able to discriminate better irrespective of the volume of water considered. Also, the reduced standard deviation in OSF indicates that the features are robust to changes in volume even with changes in the training data. During the process of swallow, the vocal folds remain closed. Given that OSF perform much better than BLF, in study design 2, Within the OS features, the features unrelated to voice source were found to have outperformed features related to voice source by around 21%. This hints that glottal excitation features like F0 and jitter and etc. fail to capture swallow sound characteristics. When the performance of individual feature subgroups within the group A features was assessed, MFCC, Rasta filtered audio spectrum, and RMS energy were found to be consistently the top performing features in all three classification tasks. The feature subgroups also outperformed the baseline features in all classification tasks. This plot here shows the performance of each feature category sorted in decreasing order of F scores after ISS2ML classification. Results of study 3. From the table, it is clear that both OS common features and OS ranked features showed almost equal classification performances, and the p-values indicate that their performance is similar to each other. On examining the features selected in the OS common feature set, they were found to be different functionals of smoothened version of loudness and its derivatives. Loudness is the sum of a spectrum computed by passing a signal through 26 null frequency bands in the frequency range of 220 to 8000 Hz with an auditory weighting with equal loudness curve with cubic root compression. This table here shows the functionals selected by the model. It can be seen that some statistics like percentile and standard deviation were selected. This could have occurred possibly since they are less sensitive to silence regions before and after the swallow sounds and hence also show higher PCC values. Even though swallow signal boundaries were unknown to the model in the OS features, the selected robust features seem to be indicating them indirectly. For example, mean and minimum functional segment lengths measure the number of subsegments where loudness is higher than 25% of its range. Features like up-level time, rise time, and curvature time indicate the time required for signal subsegments to cross a predefined threshold. All these features could be indicative of the SC2 component of their corresponding volumes of water. Therefore, to sum up, our work concludes to the following key takeaways. 1. Elimination of manual expert annotation through improved performance using OSF rich acoustic feature set and lasso selection of features that also indirectly provide cues for boundary detection. The 12 joint features of loudness perform much better than the baseline features. Finally, the lower standard deviations in the proposed features show their robustness to changes in swallow signal characteristics. From this point, we plan to expand the scope of this work by exploring swallow sounds specific features instead of generic speech analysis features and incorporating swallow signals from patients diagnosed with dysphagia so as to perform classification among healthy and dysphagic swallows. I thank you all for attending this presentation. The authors would also like to thank all the participants for participating in the swallow sound data collection. The authors would also like to thank Pratiksha Trust and the Department of Science and Technology, Government of India for their support. For any questions and suggestions, please feel free to write to us at spirelab.ee@iisc.ac.in. I would like to thank you all for attending this presentation once again. Thank you.